In the introduction section of the video on the development of the sensory organs, we will explore the intricate processes involved in the formation of the nose, eyes, and ears. This knowledge is essential for medical students to diagnose and manage related developmental abnormalities and disorders. During embryonic development, sensory play codes are formed through a series of signaling events. These play codes, which are thickened regions of embryonic ectoderm, give rise to sensory organs like the nose, eyes, and ears. The induction of sensory play codes is primarily driven by signaling molecules like FGFs and BMPs, which help specify the fate of ectoderm cells. Once induced, the sensory play codes further differentiate to form specific sensory structures. The olfactory system is important for our sense of smell. It develops through the formation of olfactory sensory neurons and their connections with the brain. Different signaling molecules and processes guide the development of these neurons, allowing them to detect various odors. In the development of the eye, different structures like the retina, lens, and cornea form through a complicated process. The optic cup, formed from ectoderm and optic vesicle interaction, becomes the retina. The lens forms by invagination of the lens play code into a vesicle, and the cornea comes from the overlying ectoderm. The lens and cornea are crucial for vision. The lens differentiates from lens epithelial cells, becoming transparent and losing organelles. The cornea develops from surface ectoderm through corneal epithelial cell differentiation and corneal stroma establishment. The retina, located in the eye, undergoes complex processes to form distinct layers and cell types. Cells originate from multipotent progenitor cells, then proliferate, migrate, and differentiate into photoreceptor, bipolar, ganglion, and horizontal cells, ultimately establishing the functional structure of the retina. In this section, we will explore how specialized photoreceptor cells called rods and cones develop and play a crucial role in our vision. We will discuss the regulation of gene expression, cellular structures, and the process of differentiation. Understanding these mechanisms gives us a deeper understanding of our sensory organs. The optic nerve transmits visual information from the retina to the brain, allowing us to perceive and interpret visual stimuli. The development of the optic nerve involves the bundling of axons from retinal ganglion cells, which are the output neurons of the retina. These axons extend toward the brain, eventually forming the optic nerve. As they navigate through the developing brain, the axons establish specific connections with their targets in the brain's visual centers, ensuring the proper transmission of visual information. The auditory system enables us to perceive and interpret sound. Its development involves a complex series of events that lead to the formation and differentiation of structures such as the cochlea and the auditory hair cells. The cochlea, which is responsible for converting sound vibrations into neural signals, arises from a specialized region of the inner ear called the otic play code. During embryonic development, the otic play code invaginates to form the otic vesicle, from which the cochlear duct, containing specialized sensory cells called auditory hair cells, is derived. The inner ear develops intricate structures for hearing and balance, including the semicircular canals responsible for spatial orientation. These canals originate from the vestibular play code, going through invagination and elongation, and house hair cells that detect fluid movements and transmit neural signals. Auditory hair cells are the primary sensory cells responsible for the detection of sound vibrations and the initiation of auditory transduction. The differentiation and maturation of auditory hair cells involve a series of intricate cellular and molecular events. Precursor cells within the cochlear duct undergo differentiation, resulting in the formation of both inner and outer hair cells. Various signaling molecules and transcription factors play essential roles in the regulation of hair cell development, leading to the establishment of a precisely organized auditory sensory epithelium. The vestibular system contributes to our sense of balance and spatial orientation. Its development entails the formation of vestibular structures, including the utricle, saccule, and semicircular canals, which are essential for detecting head movements. These structures develop from the otic vesicle and undergo morphogenesis to acquire their distinct shapes and orientations. The vestibular sensory cells, called hair cells, undergo differentiation and form synaptic connections with vestibular nerve fibers, allowing for the transmission of balance-related signals to the brain. The gustatory system is necessary for our sense of taste, enabling us to discriminate between diverse flavors. The development of taste buds involves specific steps that lead to the creation of specialized structures and the specialization of sensory cells responsible for taste perception. Taste buds derive from particular areas of the oral epithelium named taste papillae. Inside taste buds, distinctive cells called taste receptor cells show unique morphological features and express distinct taste receptors that enable them to identify various taste stimuli. Understanding the intricate process of sensory organ development is essential for medical students. This knowledge allows for the identification and treatment of disorders affecting these structures, ultimately improving the lives of patients.